Okay, well, we will go ahead and get started. Thank you everyone for joining us today. My name is Jenny Denai. I am the Senior Programs Director with the Lung Cancer Initiative in North Carolina. And this is our Power Up series that we have throughout the year. We've actually been hosting more of these lately, so thank you for joining us virtually as we do these a little different. Um, and I just wanted to go over some housekeeping before we get started. If you have questions, you can put them in the Q&A, which is at the bottom of your screen. There's a Q&A box. You can type them in there, and we will be answering those after each speaker and at the end as well. And I think that's about it. So we've got a great lineup of speakers today, so I'll go ahead and get started. Paige, if you want to go ahead and give a little introduction about LCI.
Absolutely. Thank you. I'm Paige Humble, and I'm the Executive Director of Lung Cancer Initiative. And as Jenny said, we are so excited to be able to virtually see all of you today. Our Power Up series is our series of community education across the state. And our topic today is particularly important because it's highlighting the research that we are doing right here in North Carolina and the innovations that are being utilized in this research. So we're so happy to be able to bring this to you today. And I'm just going to walk you through at a very high level a little bit about Lung Cancer Initiative for those of you who may not know about us. Today's program and our series, our whole Power Up series, would not be possible without our community partners and our supporters. So thank you to Genentech, to Celgene, and to Pfizer for your support of the Power Up series and for making today possible. Our mission at Lung Cancer Initiative is to save lives and provide support to those affected by lung cancer, really in 4 key areas: research, awareness, education, and access programs across our state. And our organizational vision is to improve life for those affected by lung cancer. And we just finished strategic planning for the next 3 years at the end of 2019. And so our 3-year strategic vision is to impact more lung cancer patients in communities of most need. And as you can imagine, we've had to pivot a lot this year in our delivery of some of these things, but I think we've learned a lot and we're excited about some of the things to come. We have also had core values from our founding, and these have remained constant and are the thread that goes through everything that Lung Cancer Initiative does. And we are the state's leading nonprofit organization supporting lung cancer and research, and we really specialize in connecting patients, survivors, and their loved ones with the medical and research community. So things like today are very, very important to us, and we continue to look for new ways to do that. And we hope that if you haven't met a member of our staff, that at some point in the very near future, you'll get to meet some of our staff members who really coordinate a lot of our efforts for the programs across the state. We also have a really dedicated board of directors, and it includes leadership from the health centers across the state, lung cancer survivors, caregivers, and community leaders. And it's really our board of directors and our network across the state that allow us to do what we do as an organization. And when we talk about these 4 key areas, I just want to walk you through at a really high level what they are. So research to date, we have funded more than $2.26 million in lung cancer research, and the majority of that is here in North Carolina, and we hope to see that continue to grow. We fund research fellows at the major medical institutions across the state, and we're actually getting ready to announce our class of 2020 fellows. So in the coming, probably the next week, you'll be able to see who those folks are. Last year, we also launched a Career Development Award, and you see Emily Harrison's picture here from UNC. She was our recipient of that award. And in 2018, we launched the Vicky Amidon Innovation in Lung Cancer Research Award. And Dr. Bishop, who's one of our speakers today, was our first recipient of that award. And again, that's another program where we hope to continue to grow. We also have access initiatives. So we have gas cards that we provide to patients to help them access appropriate care and treatment. And what you see here is this map from 2017 to 2019 of the counties where we were able to directly impact at least one patient through some of our programs. And, you know, we want 100 counties. We want to continue to do better, but we are continuing to make further inroads across the state. We also have access grants, and those are to support community partners who are facilitating some sort of access to screening, to education, to treatment, to biomarker testing. And we continue to look for partners for those grants, and they're specifically designed to reach the communities of most need. Some of our most recent access grantees, Novant Health and Cone Health, both had programs oriented around lung cancer screening, and Rebuilding Together the Triangle was oriented around radon mitigation. Also, you may have seen that this year, Lung Cancer Initiative also utilized some of our access funding to provide $250 stipends to help lung cancer patients during this time. And we had our first round of funding was in April, and we exhausted that funding very quickly and were able to help 40 patients. So we actually have another round of funding open, and it will be closing very soon for 40 more patients. And we know there's tremendous need, and we hope to continue to look for new ways like this to support the lung cancer community during this time. We also have awareness events. We always are looking for health fairs and community meetings where we can have a presence. We also believe our 5K races, our gala, they all help to raise awareness. And last year, our Lunge Forward 5K series had 90 survivors, and that was the most we've ever seen across the series, and we hope to continue to see that number grow as well. And then events like today, our education series, Power Up is our signature series where we partner with cancer centers and organizations across the state to deliver education. We provide physician education. We have free resources on our website. We have our advocacy summit. Again, we are continuing to look for ways to partner and deliver education across the state as well. And a newer focus for us has been survivor engagement. As the number of survivors across our network grows, we are continuing to look for programming that is valuable and supports survivors at all phases of survivorship. And you'll see our Survivor Steering Committee, which was just established here. There's several members not pictured, but we have representation from across the state. And this group is really helping us determine what programming is most valuable to lung cancer survivors. We also have volunteer opportunities. So if you have not volunteered with Lung Cancer Initiative, but you're interested, please reach out. You can see we've got some trainings coming up on Zoom, one on August 24th and another in November. If you're just interested in learning about all the different ways to volunteer, we need all skill levels. We need folks that are working remotely or can volunteer in person. So we encourage you to check those opportunities out. And, you know, a great way to get started is through our Lunge Forward series. Again, this year has been a difficult year for all of us. So we've had to pivot some of our events at Lung Cancer Initiative. Our Charlotte 5K actually became a virtual 5K. And it was very exciting to see people sharing pictures across social media who were stepping up to support the effort. We're going to have a celebration if you're in the Charlotte area on October 10th. And so you can still sign up and participate. We hope to be able to have it in person. That's our plan. And we would love to have you in Charlotte on the 10th. We also have our Triangle 2-mile event. It's a new format for this year for the Raleigh race. It's traditionally been a 5K, but with the North Carolina Museum of Art, which is a beautiful setting. And we're going to have a 2-mile walk and lots of new activities planned there. Again, we're really hoping for in person. And we're also going to have virtual participation options as well. And then in the Triad, we have our Lunge Forward at Center City Park on November 14th. And it's just a fun family celebration. And we hope if you're in that area that you'll sign up to participate. We'd love to have you. Our Evening of Hope, which is our annual gala, is going to be an online event this year. And we've just actually determined, excuse me, I'm losing my earphones. We've just actually decided to hold this on August the 30th. So it's a Sunday evening at 7 o'clock, and it's going to be a very fun, celebratory program, 45 minutes. We're going to have a silent auction leading up to the event, some live auction items and some entertainment. So we really encourage you to participate, and we're going to be sharing more details about how to participate shortly. Also, Our advocacy summit is coming up August 21st and 22nd. Again, you can participate in this online. It'll be an all online event. It's a great way to really arm yourself with tools to be a better lung cancer advocate. We're going to have Sharon Delaney McLeod from Walk West, who's going to talk to us about the importance of our own stories as lung cancer advocates, and a host of other breakout sessions for survivors, information for caregivers. So we really do hope that you'll take advantage of this opportunity to connect as well. And then our next Power Up series, we have one in July that's updates after ASCO. And we're going to be sharing some of the updates that were shared at the virtual ASCO conference this year, all pertinent to lung cancer, because there were quite a few advancements shared. So we do hope that you'll sign up and join that as well. And with that, I am so excited to be able to introduce to you our speakers today. As I mentioned earlier, Dr. Adler and Dr. Bishop are recipients of the Vicky Amidon Innovation in Lung Cancer Research Award. And Vicky Amidon was a beloved wife, mother, and friend who passed from lung cancer, and her family has continued to be very involved and very staunch advocates for lung cancer. And so it's our pleasure. Dr. Bishop was our first recipient of this award. And Dr. Bishop received his PhD from Duquesne University and began focusing on exhaled breath analysis at Texas Biomedical Research Institute. And in 2018, he came to Wake Forest School of Medicine as a part of the Center for Precision Medicine. And then Dr. Kenneth Adler received his PhD in cell biology from the University of Vermont. And he is the Alumni Distinguished Graduate Professor in the Department of Molecular Biomedical Sciences at North Carolina State University. Dr. Adler has published more than 150 papers related to lung cancer and airway diseases, and has received continuous long-term funding from the National Institutes of Health. And his lab has been the basis for the formation of the biotech company Biomark, which has developed a drug based on his laboratory's discoveries that have progressed to successful clinical trials in patients with chronic bronchitis, acute respiratory distress syndrome, and lung cancer. And finally, Jon Gorman is not able to join us today as a speaker, and we hope to hear from him at a future event. But Shirley Sulik is a lung cancer survivor and very, very active lung cancer advocate. And she is also a member of Lung Cancer Initiative's Board of Directors. So we'll be very, very pleased to hear her story to conclude our program today. So with that, it is my pleasure to turn the program over to Dr. Bishop.
Thank you. Let me share my screen. All right, can everybody see that screen all right? Good, okay. Well, thanks everybody for the opportunity to come and give a virtual update to the research project. So the title that, of the application was Identification and Characterization of Novel Exhaled Breath Metabolic Signatures of Lung Cancer. And so what I hope to be able to do is simplify that title to the group and explain some of the concepts through the talk today. So as an overview, I'm going to give sort of some information on the diagnostic dilemma of lung cancer. Talk about smelling diseases in cancers, go into more information about what a metabolic signature is and why it's important, and then talk about some of these concepts for using exhaled breath in lung cancer research. And I'll give a somewhat short research update because the study is currently recruiting patients, and talk about a couple other projects that are ongoing in my group that are relevant to lung cancer, but from a couple different angles that I wanted to mention. So the diagnostic dilemma, lung cancer is, like was just mentioned, is identified typically through CT or computed tomography imaging. And this is the first line of identification of a lung abnormality. And this, through the National Lung Screening Trial, which was a trial that was used to bring in patients that are high risk of lung cancer. the results from that saw an overall decrease in mortality. But what the dilemma is that 1 in 4 patients had non-lung cancer abnormalities that required further diagnostic investigation to determine if the patient had lung cancer or not. And so this was likely through a biopsy or maybe even surgery to remove the abnormality to determine if it was cancer or not. And what this does is this increases in invasive procedures as well as costs to the procedures. And so, wouldn't it be great if we can identify a non-invasive signal that would inform us if that abnormality is a cancer or not? And so that's really the basis for some of this exhaled breath research that I'm working on. And so coming into smelling diseases and cancer, this goes back into the Middle Ages time when physicians would actually smell urine from a patient and could determine whether the patient had diabetes or not. Won't go into any more details, but you fast forward to the 1970s and Linus Pauling published a paper that was developing technologies to collect volatiles off of urine samples and out of breath samples from a patient to look at what was in a healthy human urine sample or breath and trying to identify volatiles in those samples. And so the concept behind this is that you can have metabolic markers or disease markers in these fluids or in your blood circulation that could come out in your lungs and could be indicative of a disease state. And so fast forward with more technological advancements to 2020, where we're using personalized devices for performing exhaled breath collections on a patient. And so this picture is the Reciva device that's manufactured by Owlstone Medical in the UK. And I've been using this device now for a few years on various studies, and we're currently using it as part of the project to collect exhaled breath samples from our patients. And so I'll backtrack to in 2016, there was a number of articles that came out that dogs can detect cancer better than any lab tests. And so there was articles of dogs that were able to sniff out cancer. And there's actually a company in the UK that uses dogs to detect various cancers, neurological diseases, bacterial detection dogs, malaria. And so this concept is true because these dogs are detecting a metabolic signature or volatiles in the urine, and dogs have 10,000 higher abilities to smell than humans. So this is valid because these dogs are just trained to detect a signal. The dogs don't care what that signal is; they're going to get their treat if they detect it correctly. So anyway, dogs are smelling it. The concept for us is that we're using mass spectrometry equipment to determine what these chemicals actually are, or try to at least. So what is a metabolic signature? In all reality, it's a snapshot of the composition or metabolites, the composition of metabolites in a material you are studying. And for this, we're trying to determine if we can take a snapshot of the patient's breath and find information that's indicative of cancer. And so to put it in more simple terms, everybody in the room is probably likely went to have a doctor's visit and have gotten a blood sample. And then usually you go to your doctor and they give you a report that your levels are within range for your cholesterol, your HDL, your LDL, triglycerides. Or you may be out of range and you need to do something about it. But this is the concept of, a simple concept of a metabolic signature that might be indicative of a disease. And so it can use the concept of a liver tissue and your overall liver is healthy and you have your blood tests and your levels are okay. But as you move towards maybe a diseased organ or a diseased liver, you can see these metabolites or this metabolic signature change to have excess, which might correlate with the disease. And so this is sort of the concept we're starting with lung cancer. So how can we determine a metabolic signature of lung cancer? So we have to have a signature where there is no lung cancer and then a signature where there is. And so we're looking at using this study that I'm working on to get a metabolic signature in exhaled breath. And everyone knows exhaled breath is a very hot topic these days. because, as we know, we're trying to keep distance from each other. And this is a representation of a simple breath. You can have a lot of information and metabolites coming out of the breath. And so is there a change between a healthy, non-cancerous lung and whether there's a tumor there or not? And so our study design is a little bit backwards, where we're taking patients before surgery, taking a breath sample, and then they go through their tumor removal, and then we do another collection a month later. And we're looking at the breath profiles in both and seeing if we can find differences. So a little bit of background of the breath collection process, because I'm sure everyone is curious, given the current situation we're all in. There's a volatile phase. So there's 3 phases of breath. There's a volatile phase, and these are the volatiles that I have been talking about up to this point. I'm using, like I mentioned, the Reciva device to do this collection. And what I like about it is it's a portable device that I can walk into the clinic, get a breath sample from the patient, and walk back out in 5 minutes. And so this is my setup. On the right here, you see there's a setup. It can run off your Surface Pro. You have my trusty glass head I use for doing blank air collections. And I can pipe right into the medical air in the hospital to do the collection. On the left, you can see sort of an output of the controls of the system. Now, the company that is producing this device is pushing the concept of a breath biopsy. So we have a tissue biopsies that we're very familiar with, but now we can do a breath biopsy. We can collect various fractions of the breath and use it to hopefully identify a metabolic state of the patient. What I like about this device too is it monitors the patient, it monitors their breathing rates, and it can turn on and off pumps at particular parts of the patient's breathing rate. And so you can collect just the time the patient's exhaling, which is good for concentrating a signal. And so this is, a technological advancement that's helping my research. Second phase, aqueous phase. This is collected, you can think of the concept of in the winter. I grew up in New York, and we had a lot more winter than we do here in North Carolina, but very familiar to seeing my breath when I walk outside in the winter. That vapor that's freezing when it hits the cold air is your aqueous phase of your breath. And that's been, the subject of conversations with everything going on these days is that's not only carrying potentially viruses, but carrying information about your metabolic state. And so we are using the R2, which this is somewhat of a pixelated image from their the company's website, Respiratory Research Incorporated. But what we do is this, the patient breathes through a mouthpiece, and as they breathe, they pull in air from the bottom of the device, and as they exhale, it goes out the top of the device, and we have a frozen metal sleeve around the device that is concentrating the breath condensate into the tube. And at the end of the collection, the frozen breath condensate melts, and we're able to collect it and freeze it down for analysis. The last phase, and I will say I made this slide before in the COVID-19 situation, but the best way for collecting aerosols is your mask. And so this is the third phase. It's in combination of the aqueous and particulates that are expelled as you breathe, as you talk. And so I don't use this part as for my research, but it is the third phase of breath that's relevant for everything going on. So after receiving the award, and during writing the award application, I put together a team here. I had a research team, which was people involved in my group to do the research, and the lung DOT team, which is coordinated by Dr. Petty, and a clinical team of pulmonologists and surgeons that were going to help me carry out this research to recruit patients and then identify the correct population of patients that we need to. With a study objective to using these advanced technologies to profile lung cancer in non-invasive samples prior and post lung cancer surgery. And so a research update, somewhat very simple, is that we've had IRB approval for some time now, and we're actively recruiting. We have been shut down from recruitment for the since about March. But we're slowly getting back to the point where we should be able to start recruiting soon. Part of the dangers of collecting exhale breath are pretty evident to everybody now. And so what we're having to do is implement safety precautions for myself and my staff so that everyone's safe and we're not putting ourselves at increased risk by performing these collections. And so a lot of it involves just what you would expect, wearing personal protective equipment and doing it in specialized rooms that have proper ventilation. And then properly freezing down the samples as well. And so to give everybody sort of a little bit of an insight of what a breath sample looks like on the equipment that I run, this is a chromatogram of a breath sample that we've collected on this study. And everyone is looking at it, just sees a bunch of lines, but I see data, and you can look at one peak here in particular. And the hard part about doing this on a presentation is you can't actively zoom in, but you can take screenshots and zoom in. And what this peak is identified here is benzaldehyde, and you can zoom in farther, and then it finally actually looks like a peak. And so what the software does, and a lot of this is all done by software, is identify these peaks and match them up to databases of compounds, which can then be made into a list. So you have a list of peaks from metabolites that are identified in a patient's breath. The next thing we need to do is compile them across all the patients. So we have patient 1, patient 2, patient 3, patient 4, and so forth. And when all these patients have then gone through surgery, and then you do another collection. And then all of this data is aligned across itself, and we were able to find peaks that are relevant to the various groups. And so the idea that was what we're going for is, can we identify a metabolic signature pre-surgery state where a tumor is present, and then post-surgery. So we're going to have some overlap there. We're hopefully going to have overlap because the patient's not changing. And then hopefully we have metabolic signatures that are present before, or maybe absent before, and then present after, or maybe absent after. So the idea is to look at all these combinations of metabolic signatures and determine if we can identify some potential targets. So with that, I'll move into a couple other studies briefly. One is relevant because it was initiated by Ryan Hughes, who received the 2019 Lung Cancer Initiative Research Fellow Award. With Michael Ferris and myself, we've been mentors to Ryan. I think Ryan is soon to become leave residency, but he's staying here at Wake. So we're going to continue to collaborate together on this project to move it forward. And the idea of this project is using exhale breath to assess lung injury. And so you think, well, lung injury and lung cancer, where does that mix? The idea is that some patients are going to go for radiation treatment, and the radiation is going to kill the cancer, but it's also going to impact healthy lung tissue. And so within this population of stage 3 lung cancer patients, about 20% of these patients will develop symptomatic pneumonitis. And what this is severe inflammation that will have to be treated, usually with a steroid treatment. And you might say, well, why don't we give everybody steroids and then we might stop it all? And that can't be done because some patients might have adverse effects to the steroids. So they have to wait till they see symptoms and then they start treatment. So the idea of the study is really, can we find a signature in the breath that can predict which patients will develop pneumonitis and which ones will not? And then can we put those patients that are higher risk on a steroid treatment early to prevent their symptoms from their lung cancer treatment? Because right now there's no clinical tool to predict pneumonitis development. There's been some work in blood, but we're looking at the lung cavity as closer to the disease and closer to the injury as maybe a relevant way to predict this pneumonitis development. So that's one study that's ongoing. Again, we've been impacted by the labs being shut down, but we're actively trying to get it back up and going right now. The other study is related to vaping. And this is a project that I got involved with about a year ago. It's developed over time, and I've pulled some slides from a previous talk, and we developed this Project Marvel. And the acronym, this group likes to use acronyms. It's a multidisciplinary assessment of risks from vaping during early life. And the research focus was focusing on adolescents because a lot of kids are vaping. And this is in collaboration, right around the time that I joined Wake Forest, the Tobacco Control Center of Excellence was started within the Comprehensive Cancer Center here at Wake Forest. And this group is working to expand knowledge to reduce the burden of tobacco-related disease through research, education, training, treatment, and implementation. And I joined the group about a year ago, like I said, and I have come into a number of great collaborations. And what we're doing is trying to get some projects off the ground. And for the sole fact that here in North Carolina, we have, and this is data from 2017, but increased number of kids that are still smoking cigarettes, and larger numbers of kids that are vaping compared to the US average. And these numbers are likely greater at this point. And so our group took an initiative to look at brain health, cardiovascular health, and respiratory health, and looking at the detrimental effects of vaping on health in these adolescents. Because it's really not known if vaping is going to develop into increased risk of lung cancer, but it might. And so I co-led with Dr. Ortega, the respiratory health component of this project. And it's an interesting project that we're continuing to move forward to see if we can help mitigate the impact of vaping on the youth population. So with that, I'll give a brief acknowledgement of the various teams I've been working with over the last 2 years. And especially like to thank the Lung Cancer Initiative for their support through the Vicky Amidon Innovation grant. It's been very helpful to sort of get me off the ground here at Wake. So thank you, and can take any questions. This is one of my graduate students, Justin, who was training on the equipment. So.
Well, thank you, Dr. Bishop. That was really interesting. And I've heard you speak before, but I still learned some new things. It's always great to hear you talk. We will go ahead and move into Dr. Adler's talk, and we will answer questions at the end, time allowing. So Dr. Adler, if you want to go ahead and get started and share your screen.
Okay, thanks very much, Jenny. And it was a really nice introduction you gave me, so I can skip my first slide. Yeah, I'm a professor at North Carolina State University. I've been there for, I guess, over 30 years now. And I was involved in this development of this startup biotech company called Biomark. And we just finished a phase 2 clinical trial in non-small cell lung cancer. The patients that were involved in this trial were patients with stage 3 lung cancer. And this was a trial to evaluate, as it says here, the safety and efficacy of this drug. What makes this drug different is that it's given as an inhaled aerosol. You see it's called inhalation solution. It's actually taken sort of like a asthma drug with an inhaler. And so it was an interesting and a different type of drug. So the actual trial, I'm going to present this talk a little bit differently maybe than other talks. I'm going to start off with the results of the clinical trial, sort of like reverse engineering, and then go back into how I think the drug works, which was the basis for the grant I got from the Lung Cancer Initiative. So this was a trial that was done in different centers, 10 different centers, in patients that were not receiving immunotherapy. Everyone knows that immunotherapy is the probably the latest type of treatment these patients were not candidates for immunotherapy. They were receiving chemotherapy at the time. The chemotherapy they were receiving was a combination of pemetrexed and carboplatin given intravenously. And the study was conducted, as I said, in 10 clinical centers. There were 60 patients involved. See the ages and both sexes. They were randomized, so 30 subjects received the drug and concentration of 125 milligrams. BID stands for twice a day, so they were inhaling it twice a day in conjunction with their chemotherapy. It wasn't to take the place of the chemotherapy, but rather than to get to be in addition to chemotherapy. And if I didn't say it, SOC stands for standard of care. The additional 30 subjects were sort of the controls, placebo group, if you will, and they received only the standard of care chemotherapy. The treatment period was for 3 months and then a 9-month survival follow-up period. So I want to present the exciting results from this trial. Before I do that, let's familiarize ourselves with some cancer treatment terms when oncologists look at progressive treatments. Usually they do the CT scans and looking at the tumors to see if a drug is working. So PR stands for partial response, and that means that there's a greater than a 30% decrease in the size of the primary tumor and no further metastases. SD stands for stable disease, which is no further metastases and no growth of the primary tumor. The overall response rate, abbreviated ORR, is a combination of patients with stable disease or partial response. On the negative side is what's called progressive disease or disease progression. And those are patients with more than a 20% increase in the size of the primary tumor and or additional metastases. So let's look at the results at 2 points. One is an interim evaluation, which was done at 3 months, and next one will be the 12-month data. So starting from the left of this slide, looking at this graph, we see the patients with placebo or standard of care in blue, patients with the drug in green. So this is at 6 weeks looking at partial response. That's a decrease in the tumor size, substantial decrease in the tumor size, as well as no further metastases. And you can see that there's a big difference between patients at the drug versus patients just on placebo. That's at 6 weeks. This is a statistically significant difference. We get to 12 weeks or 3 months, and we still see a big difference between patients on the drug in the green there and patients on placebo. About a 25% increase in partial response at 12 weeks. If we go to the negative side, the disease progression, we see that patients with placebo had a much higher incidence of progressive disease or disease progression than patients with the drug, 16.7 to 6.7%, and this was statistically significant. And to achieve statistical significance with this small a population is pretty remarkable. So moving on to the next slide, these are the 1 year data. Look at the results at 1 year. So we're looking again at the graph on the right. The first set of bars here is survival. So at the end of the trial, 30% of the patients, which was 9 patients, who were taking the drug were still alive versus 7 patients, 23% on just the standard of care of the placebo. And what's amazing, if you look at these second set and the third set of bars, all the patients who were still alive at the end of the 1 year period, which was 30% of the patients, showed what's called an overall response rate, which is either stable disease or partial response. There was no further metastasis, no growth of the primary tumor. This only occurred in 13% of the, just let me clarify the numbers here. These are the percentages of the 30 patients that started, just to be clear. About a little less than half of the patients that were still alive on placebo had the overall response rate, the stable disease or partial response. And if we look at the progressive disease, none of the patients who were still alive at the end of the period, 9 of those patients, none of them had progressive disease versus I think it was 3 patients, which would be 10% of the 30%. And again, these numbers here did reach statistical significance. I'd also point out there are no side effects with this drug. And I think we're all familiar that a lot of cancer treatments have pretty devastating side effects. There were no side effects associated with this drug. Okay, so we know that it works. And we know that it worked in a clinical trial. Question is, how does it work? And I said before, this is the basis for the studies that I'm doing, being funded by the Lung Cancer Initiative of North Carolina. So the study, the data I'm going to show you now, the pictures, all have to do with cancer cells that are grown in chambers or in petri dishes or in test tubes. But we did a lot of studies with animal models of human lung cancer and found essentially the same results we saw at the clinical trial. The drug is very effective in vitro, but to look at the mechanisms, to look at how this drug works, we now turn to cancer cells that are grown in petri dishes or chambers or test tubes and try to see what's going on. So these are cells, cancer cells, human lung cancer cells, very aggressive, that are grown in what's called migration chambers. This is a model of cancer metastasis. We put the cancer cells on the bottom of these filters and they migrate up to the top of the filters where they become visible. So what we're seeing here, each of these little red areas here represents a single cancer cell. So we have a highly metastatic group of cancer cells here under the control. These are untreated cells. They're treated with, moving to the right here, what's RNS is just a control peptide for the drug to show that you can't just throw anything in there and get an effect. And then as we go with our drug here, starting at 50 micromolar and moving up to 200 micromolar, as the dose increases, we see less and less migration or metastasis of these cells in these chambers. So you can see that the drug pretty much stops these cancer cells from moving. Okay, so let's look at why this is occurring and what this drug is and how it works. So these are the same cells, same cancer cells, and these are stained with this green stain here, which is a stain for a protein called actin. Actin is the protein in the cells which causes them to move. And you can see in these normal cancer cells, a very, very aggressive human lung cancer cell grown in, again, in these migration chambers, all the actin is on the outside of these cells. This is a cross section through these cells, so they'd be almost a cobblestone appearance. All of this actin is on the outside, and that is what you see in cells that are highly motile, that move a lot. After treatment with the drug, and these are the same cells, same magnification, same brightness, we notice several different things that this drug does to the cells. First of all, it causes the cells to flatten. Rather than being cobblestone or round in appearance, they are now plate-like or just flattened out on the surface. All the actin that we saw on the outside of the cells, the untreated cancer cells, now moves to the inside and forms what are called stress fibers, which are very characteristics of cells that don't move. And even more amazing, all these little dots here, the end of the actin filaments, are what's called focal adhesions. And the purpose of focal adhesions on these cells is to grab on to the substrate, what these cells are growing on, and keep them from moving. So basically, treatment with that drug caused these cells to change from cells that are highly motile, like you see in aggressive cancer cells, to cells that are basically structural and don't move. Okay, so let's get back to Bio 11006 to see how it could be working. So 11006 is identical to an important segment of a protein called MARX protein. What is MARX? MARX is an acronym for myristylated alanine-rich C kinase substrate. It's a protein that's found in all cells. When MARX gets activated, and I don't want to get into too much of the scientific weeds here, but just say when it's activated, it adds a phosphate group, and that's then known as PMARX. So PMARX, phosphomarx, phosphorylated MARX, is the what happens to MARX when it gets activated and causes cells to move and grow. And amazingly, cancer cells have very high levels of phosphomarx, PMARX. And because of these high levels of PMARX, this leads to tumor growth and metastasis. Just to show you how prevalent that is, these are biopsies from a human lung cancers. These are tissues looked at under the microscope, and they're stained for PMARX. So the darker the section, the more PMARX you see in the cancer cells. So move from the top left. These are staining controls on an A, just to show that it doesn't, that it works. Doesn't stain everything. B is just a normal human bronchus or the airway, showing very little, if any, staining for PMARX, what we expect. As we move from C, D, E, and F, these are stage 1, stage 2, stage 3, stage 4 lung cancer. And you can see that as the stage increases, the staining becomes more widespread and more intense. So the further along the cancer, the more PMARX it contains. So we believe that MARX and the PMARX concepts are key to lung cancer metastasis. First question is, why is there more phosphomarx, PMARX in lung cancer cells? Well, in normal cells, the PMARX doesn't stay around. It does what it's supposed to do, the cell, then PMARX gets changed to MARX. It loses the phosphorylation by action of an enzyme called protein phosphatase type 2A, abbreviated PP2A. So PP2A in cells keeps MARX from being continuously phosphorylated in normal cells, non-cancer cells. But why can't cancer cells get rid of PMARX like normal cells? Well, the answer is lung cancer cells contain a protein called CYP2A. What does CYP2A stand for? It stands for cancerous inhibitor of PP2A. CYP2A blocks PP2A activity in these cancer cells. As a result, they have high levels of PMARX, and that leads to tumor growth and metastasis. Well, can we modulate the PP2 activity in these lung cancer cells, and will that affect metastasis? We can look at this because fortunately there are some reagents that we can use to target PP2A. We can increase PP2 activity if we add to cells a drug called FTY 720. This increases PP2 activity, PP2A. We can decrease PP2 activity with a drug called fostroiesin. These are specific drugs here. So if we add these to human lung cancer cells in migration chambers and how they affect migration, which is a model of cancer metastasis, what would happen? FTY 720 would activate PP2A, cause a dephosphorylation of MARX. Less PMARX should inhibit migration. On the other hand, fostroiesin, which blocks PP2A, results in more phosphomarx, and that should enhance migration. Sure enough, when we look at these migration chambers, these are cancer cells, we see that's what happens. So at the top left here are cells that are untreated, cancer cells. You can see a lot of them move to the top of the chamber, showing they're very actively metastatic. And here, 1, 2, 3 to the right and in the middle we're adding FTY720, and it dephosphorylates, it activates PP2A, dephosphorylates marks, and you see less and less migration / metastasis as the concentration increases. The bottom two squares here show the opposite. When we add the phosphotriacin, this blocks PP2A, get more P phosphomarks, P marks. This enhances migration, as you can see. So PP2A marks seem to be key to the whole concept. And we believe that what we're studying is how bio-11006 works, and we think it activates PP2A in these lung cancer cells, and as a result, results in dephosphorylation of marks.
Dr. Adler, I'm sorry to interrupt. We've got about 2 more minutes.
That's all I got.
Perfect.
So,
thank you.
This is looking at human lung cancer cells, and you can see when we add bio-11006, it enhances PP2A activity, similar to the control here, the highest concentration of FTY720. So we believe it blocks the function of phosphomarks by causing dephosphorylation of marks in these cancer cells, and thereby overcomes the effects of CYP2A and does so by activating PP2A in the cells. So it changes phosphomarks to marks in these cancer cells. So just conclude, high phosphomarks levels in lung cancer cells correlate with metastasis and tumor growth. 006 activates PP2A, decreases marks phosphorylation, inhibits metastasis, and it has efficacy in reducing progression of lung cancer in patients when administered as an inhaled aerosol together with standard of care. So thanks very much for your attention, and hopefully I can get back to the lab and very soon and continue doing these experiments. So thanks very much for your attention.
Thank you, Dr. Adler. That is really exciting information there. We hope you can get back soon too, continue doing that great work. We will go ahead now to Miss Shirley Sulik, who is a lung cancer survivor. Shirley, will you go ahead and share a little bit about your story? Oh, hold on. You're on mute. There you go.
Can you hear me now?
Yep.
All right. My story is in 2014. I received this magazine in the mail. It was from Duke University. And the magazine was advertising lung cancer screening. And I looked through it, I read through it, and I said, you know, I may be a candidate because I had smoked. I had lived in Poland where there was a ring of soot around the sky. I'd come home at night and this blouse would be gray. I also lived in Moscow where the Russians, the KGB, was beaming microwaves at me. So I said, I need to be tested. So I called Dr. Garth's office and made an appointment, went in, and she said, usually your CT would light up like a Christmas tree if you have cancer. She says, but yours is a little dull. So she says, I'm going to send you over to a pulmonologist, and I went to Dr. Wong, and Dr. Wong says, well, your breathing is not what it should be, but the only way we're going to find out if you have lung cancer is to do a biopsy. So Dr. David White, did the biopsy, and he came in and he says, I want to send your tissue off for genome testing. He says, I don't think you'll have the mutation, but I'm going to send it anyway. I said, excellent idea. So I went back to the clinic maybe a couple of months later, and I did have the mutation. I had EGFR 19 positive. So I went on Tarceva. And I didn't agree. I had rashes all over my body. And I think I was on it for about a year and a half. After I came off Tarceva, I went on Tagrisso. Tagrisso, and I agree. I had no side effects whatsoever. I am a 6-year cancer survivor, but
Hold on, you just muted yourself, Shirley. There you are.
I'm going to make you a poster child. So I guess I was the first person she had received who just walked in and said, please test me. So I've been working, she introduced me to Lung Cancer Initiative, and I've been working with them, I think now for about 5 years. A truly excellent organization, and I'm glad that I found them. So that's basically my story. Oh, I was diagnosed with stage 4 lung cancer. I didn't have a solid tumor. I had ground glass nodules in both lungs. And in July of 2019, I was categorized as being in remission. So that's my story. I have an appointment at Duke on the 22nd for blood work, and then on the 25th with Dr. Gart. So anxiety is about to set in, even though I've been diagnosed as being in remission. So that's my story.
Thank you for sharing, Shirley. It's good to see you and good to hear your story as always. We will definitely be sending some good thoughts your way for your scan.
Oh, great, thanks. It's scary when you're going into the hospital and you get your brain scan, your chest scan, you get your whole body scan. So therein lies the anxiety.
I bet.
Yeah.
It doesn't look like we have any questions. If anyone has any questions, we'll wait a minute or so if you want to put it in the Q&A, or you could even put it in the chat box if you wanted to. We can just wait a minute to see if anyone has any. And if not, or if you have questions later, just email me and I can pass it along to our speakers as well. I'll just wait a couple more seconds.
So I had a question in the, Colleen asked a question in the Q&A. I don't know if you can see that.
Oh yeah, go ahead if you want to.
I had answered it because I didn't know if we would run out of time, but are
Okay.
there any diseases that have diagnostic tests through breath tests, or are there other research you're aware of working on developing breath tests to diagnose other diseases? Yeah, there's hydrogen breath test that can be used for looking at overgrowth of bacteria in the GI, and then a lot of the pulmonologists use exhale breath nitric oxide to help diagnose asthma. There's a lot more research than in clinical testing, and I just named a few that were kind of going off the top of my head, but gastrointestinal diseases, diabetes, metabolic disease, Alzheimer's disease, liver disease, colorectal cancer, asthma, COPD, bacterial infections. So these are all publications that I've seen. I've likely missed a few, but there's a lot of research going on in this. And even in lung cancer, I mean, Owlstone Biomedical, which is the company that's putting out the device I'm using, has initiated huge clinical trials. So I am a small boat in a big sea of breath research. So.
That's great, thank you. And just so everyone knows, I know we're running over, but if you'd like to stay, I've got a couple more questions that we can get answered here. Miss Shirley, are you still on Tagrisso?
Yes, I am.
Okay, that was one of the questions. Dr. Adler, are there plans for clinical trials of your drug in larger groups? Oh, let me
I can answer that question and the next one.
Okay, good.
Putting together a protocol for a phase 3 trial, which would be, rather than in combination with chemotherapy, would be in conjunction with something like Keytruda or a PD-L1 inhibitor. It's a very good question, and the answer is yes, we are in the process of developing those.
That's exciting.
Plans for protocols to do that. The next question says about EGFR. I don't know the specifics of the individual patients who are on the trial. I would imagine that it would make no difference if it's on, EGFR mutant or not. I would think so. A lot of the cells that we do work with in vitro in chambers are EGFR resistant or EGFR sensitive types of cancer cells. So my answer would be, it probably would be appropriate treatment for that.
And I think you pretty much already answered this, but would it work in conjunction with a PD-L1 inhibitor?
Yeah. That was, yeah, I said that we believe it would, and we are planning to do a phase 3 trial in conjunction with a PD-L1 inhibitor.
Okay. I think that's, there's some in the chat here. Let's see, are there questions there or just? Okay. Well, I think that's about it. Thank you everyone for your time, and thank you to the speakers. Paige, do you want to say anything?
No, I think you said it. Speakers, you did a fantastic job. Thank you so much. Thank you to everybody for tuning in and for your questions. And we just look forward to seeing you all at the next one next month. Thank you.
Thank you all so much.
Bye-bye.
Bye.